Hello, and thank you for joining me for this webinar on expression vectors, how to choose or customize vectors for gene and protein expression. By way of introduction, my name is Rachel Spear, and I'm a technical writer and marketing specialist here at GenScript, focusing on our molecular biology services. I did my PhD work in the neuroscience program at Weill Cornell, and throughout my research programs there, I used a variety of different expression constructs to express recombinant fusion proteins for reporter assays and for biochemical analysis. I see that most of our attendees today are using E. coli or mammalian host cells, so I'll focus case studies on those areas, but we will touch briefly on other expression hosts, as well as on delivering expression constructs in vivo. If you have a question at any time, please type it into the chat function. After the presentation, I'll read the questions that have been submitted and do my best to answer them. This webinar, including the Q&A, will be archived on our website for you to view on demand in the future and to share with colleagues. My goal today is to introduce you to expression vectors. First, what are the critical features of vectors and how do you read a plasmid map to identify the features that matter to you? I've tried to make this more approachable than some other resources out there by organizing the content around the stepwise goals that you want to accomplish and then describing the tools available to help achieve that goal. Second, we'll look at the most popular expression vectors for bacterial and eukaryotic expression systems and discuss the pros and cons of readily available commercial vectors. Then we'll look at some case studies where researchers have decided to customize an expression vector. We'll look at what they wanted to achieve, how they made their modifications, and how successful their efforts were. Finally, we'll take a step back and look at the big picture of all the factors that influence heterologous protein expression so that you can see how vector selection fits into your larger workflow. And I'll introduce you to a number of solutions that GenScript offers to help you get better protein expression for your work. So if you're one of the many researchers who have used only a single expression vector simply because it's the one you're familiar with and it's easy to make new constructs by altering the gene insert in that one vector, my goal for this webinar is that you'd come away feeling confident to tinker with the components of your vector or to try building constructs in different vectors in order to streamline your workflow or to get better expression results. So let's get started. What are expression vectors? Put simply, expression vectors are plasmids that carry cargo, that is, insert DNA, into cells and allow the cargo DNA to be expressed, that is, transcribed into mRNA, and in most cases, translated into protein. Most of the time, your goal is to express a heterologous protein. So your cargo will be DNA that encodes a protein, and you'll need your expression vector to facilitate efficient transcription to mRNA and translation to protein. In other cases, your cargo might encode a functional RNA. In that case, transcription alone will be sufficient. For example, if your DNA cargo encodes short hairpin RNA that you're using to knock down endogenous gene expression, or if you're doing CRISPR-mediated genome editing and you need to introduce guide RNAs into the cell. But the most common use for expression vectors is to express protein, so we'll focus on that today. Several vector components are necessary to achieve all of the steps leading to high-level expression of soluble, properly folded protein. So depending on the goal of your experiment, you need to decide whether you want your gene of interest to be expressed from an extra chromosomal plasmid or whether you want to integrate your gene into the genomic DNA of your host cell. If you want to express a protein at high levels in order to purify it, or if you want to do a reporter assay where overexpression driven by a strong promoter is appropriate, then expression from a plasmid is easy and can be ideal. However, in some cases, you may want to integrate your expression cassette into the host DNA. Using viral vectors can be easy and highly efficient, but viral-mediated gene transfer is not targeted. That means that genomic integration may occur anywhere in the genome, so there's some chance you may be disrupting some endogenous gene or regulatory region. A way to get around that potential problem is to take a targeted approach so that you integrate your expression construct in a specific known location in the host genome. This has been done using homologous recombination, but it recently became a lot simpler and a lot more efficient with the development of CRISPR-based gene editing. So compared to older technologies like zinc finger nucleases or TALENs or CRELOCs, CRISPR has several advantages. The constructs are easy to design, the efficiency of knock-in is much higher, the targeting is very specific, so you don't have to worry about off-target integration having unintended consequences, and there's no additional foreign DNA introduced at the targeted sites. So and for most cell types, you can get high efficiency without using viral delivery if you want to avoid viral vectors for biosafety reasons. So targeted genome editing is definitely the preferred approach if you want to study gene function under the control of endogenous promoters, or if your assay requires that you don't disturb signaling cascades by introducing an overexpressed variant that's going to outcompete endogenous proteins for binding partners. In these cases, instead of overexpression from a plasmid, you should consider a targeted knock-in strategy. We'll return to this idea in our case studies, but for now, let's dive into examining the plasmids we're going to use for expression constructs. So reading a plasmid map, it may seem confusing at first, but just like reading scientific papers, it's a skill that you develop over time as you become more familiar with the terminology that's used. So the rest of this section will hopefully jumpstart your plasmid reading skills. Plasmids are circular pieces of DNA, so maps usually look like a circle as shown here. The value of a map is to provide a quick and easy summary of the sequence features and to show the relative position of those components. Position within the plasmid is communicated by numbering each nucleotide, starting at the origin of replication. The ordering of components is critical in some places and not so much in others. For example, the location of antibiotic resistance genes like AMP and NEO shown on the map here are usually historic and not necessarily optimal. You can sometimes get better performance by moving these pieces around, but that's not often done. However, the position of open reading frames relative to the promoters that drive their expression is in fact critical. And on a smaller scale than is visible here, it's critical to ensure that when you clone in your gene of interest, it gets inserted in the same open reading frame as any fusion tags, such as the DYK tag or flag tag that's shown here. To get that nucleotide level resolution, you'll need to consult the full sequence. A map should always be accompanied by the full sequence of the plasmid. And, caveat emptor, plasmids are often shared and repurposed without sequencing the entire backbone, which means mutations can accumulate. And those may or may not impact your experiment, but just be warned, if you get two clones from two different sources, even if they both say that they use a pcDNA 3.1 backbone, they may in fact have slight differences in the sequence that won't be apparent from the map alone. So as you work with plasmids, you'll almost certainly want to start using a software tool that allows you to analyze and edit plasmid sequences and maps. There are many such tools available. Some are free and some are not. I personally have used SnapGene and APE, which are both free tools, and those have worked for my needs, but other people have different preferences. If you have an experienced molecular biologist in your lab, it might be easiest to quickly get up to speed on using a tool by watching them. Or if you have more time to explore different tools, that may be worthwhile. In addition, when you don't need to do a lot of analysis to a plasmid sequence, but you just have a quick question, such as what are the unique restriction sites in this sequence, there are some web-based tools with more focused functions, such as the NEB Cutter tool that are widely used. So now let's dive in and think about all the things you want your plasmid to be able to do and what components it needs to contain in order to achieve those goals. So I know this looks like a very busy slide. I've organized this to serve as a reference that you can come back to. And I want to sort of focus on what our goals are and what our workflow is. So we'll go through each of these steps in more detail on following slides. So don't worry about digesting it all at once. So what's the first thing you need an expression vector to do? Well, you need to be able to insert your cargo and then verify that it's been inserted accurately. So this is most often done with the help of restriction enzymes, which recognize specific cleavage sites. So the region of the vector where you will insert your gene of interest contains a lot of different restriction enzyme recognition sites right in a row. This is called the multiple cloning site, or MCS. There are other cloning methods which require special vectors containing recombination regions or other adaptations, and I'll discuss those a little bit later. So then after you insert your cargo, or really any time you acquire or modify a plasmid, you'll want an easy way to verify the accuracy of the insert sequence. So most vectors include common sequencing primer sites such as T7 and M13, flanking the MCS. Most DNA sequencing services will offer primers for those sites at no cost, whereas if you need a custom primer, it'll cost more. So you might look for 5' and 3' primer sites just for convenience for sequence verification. Next, you're going to want to insert the plasmid into cells. For certain delivery methods, you need to ensure that your plasmid backbone is compatible. For example, for viral-mediated delivery, your backbone needs to contain components required for viral packaging. Then you need a way for the plasmid to initiate its own replication within the host by recruiting transcriptional machinery proteins. This is called an origin of replication. And you need a way to identify cells carrying the plasmid so that you don't have a mixed pool of transformed and untransformed cells. This can be accomplished with a selection marker, such as antibiotic resistance, which will allow only the transformed cells to survive. Or you could use a screening marker that lets you visually see which cells are transformed and which aren't, for example, through blue-white screening. Next, once you've got the cells that are carrying your plasmid, you need to transcribe mRNA from the plasmid. This relies upon a transcriptional promoter. Depending on your experiment, you may choose a constitutive promoter or an inducible promoter. The promoter region includes the sequence to which polymerases will bind, as well as operator regions where enhancers or repressors may bind. And of course, you need to dictate where transcription should end by including a terminator sequence. The next task is to translate mRNA into protein. So just like with endogenous mRNAs, your expression construct needs to include a ribosome binding site and a start codon to allow the ribosome to initiate translation, and then you also need a stop codon to end translation. To promote proper folding of nascent protein, your first thought might be, well, if there's a chaperone protein that assists in the folding of my target protein, then I could co-express a chaperone. That's true, but not all proteins have known chaperones or single chaperones, and there are several other approaches that you can take that may be more powerful and widely applicable. For example, you can add a solubilization tag to your expression construct. Or you can custom design a synthetic ribosome binding site that will slow down the translation initiation rate to allow enough time for folding to occur. And you should consider codon optimizing your open reading frame, which can adjust the rate of translation elongation, again, either to speed it up to maximize protein yield or to slow it down in order to allow enough time for proper folding of the nascent protein. We'll discuss all these in more detail later. Finally, to visualize your protein in a gel or a Western blot, you may want to add an epitope tag, which is simply a small molecule, typically a short peptide, that is readily bound by an antibody and which is not expected to alter the structure or function of your target protein. Some epitope tags, such as His6, are especially useful for purifying proteins by using a column or other form of affinity purification. You can design an expression construct such that the epitope tag can easily be removed after purification before you go on to characterize your target protein. In addition to using an epitope tag, you might want to use a reporter, such as a fluorescent protein like GFP. So now we're going to look at each of these components one by one, starting with your cloning method. As I mentioned, most of the time you'll want to use restriction enzymes to clone your cargo into the vector. As illustrated here, you'll simply digest the ends of your insert and your vector in order to form complementary overhangs, and then ligate them together. Using a double ligation approach can ensure that your insert has the correct orientation. These methods have been used since the 1970s, and they continue to be widely used because they are simple, inexpensive, and efficient. And there are a large number of expression vectors available that include an MCS to accommodate this strategy. Other methods are available. For example, if you need to not have any restriction sites flanking your inserts, then you could use type 2S restriction enzymes, such as BSA1. These allow seamless cloning because BSA1 will cleave outside of its recognition site, removing the recognition site during ligation. This approach does require PCR extension to add the type 2S recognition site to the ends of your insert. Another option is single base overhang ligation. This is popularly known as TA cloning. It takes advantage of the fact that Taq polymerase leaves an extra A nucleotide at the end of PCR products. So if you have an extra A on your PCR product and then you add an extra A onto your vector, you can ligate them together with that single base overhang. Other variations on this use topoisomerase to ligate PCR fragments into vectors using either overhangs or blunt ends, in some cases with the topoisomerase attached to the vector. Another approach is ligation-independent cloning, or a variant sequence and ligation-independent cloning, SLIC. This also requires the addition of overhangs, but it doesn't require a ligase enzyme, as the name suggests. And then finally, recombinational cloning, again, requires the addition of flanking regions that will be recognized by a site-specific recombinase. This strategy is used in gateway cloning and in Crelox-mediated recombination. This method makes it easy to shuttle inserts between different vectors without the concern of having to avoid internal restriction sites, because it's very unlikely that your site-specific recombination sites will be found internally in your cargo. However, these methods are more expensive and they require a longer setup time. In any event, once you've got your cargo inserted into your vector to form your complete expression clone, it's time to consider how you'll deliver that plasmid to your cells. So this chart is not exhaustive, believe it or not, but it is meant to illustrate two things. First, your delivery method depends very much on your host. And second, for any type of host organism, you have multiple options for delivering expression constructs. So if you're not seeing good efficiency with one method, it's worthwhile to consider other methods. You'll note here that we use the term transformation when we're talking about delivering expression constructs to bacteria or yeast or plants. But in animal cells, the term transformation can also refer to a cancer process. So to avoid confusion, we use a different term, transfection. And the last part of that word, transfection, gives a nod to the fact that mammalian expression vectors utilize some components from viruses which mimic infection. But transfection does not actually use viruses. It instead uses lipoparticles or other methods to get naked plasmid DNA into cells. There are several viral systems that have been adapted for use in gene delivery. That is, they've been altered so that they can't replicate inside cells or produce pathogenic viral proteins, but these viruses are much more efficient than non-viral delivery methods. Efficiency may not matter much if you're working with a robust cultured cell line, but when you're working with primary cells or animals, efficiency can be critical. Lentiviruses in particular have become a preferred method for gene delivery to primary mammalian cells because they enable genomic integration even in non-dividing cells, such as neurons. Adenovirus typically does not integrate into the genome, but adeno-associated viruses may integrate, and both of those are typically used for in vivo delivery into live animals. And again, on terminology, when you get your expression clone into a host via a virus, instead of calling it transformation or transfection, we call it transduction. So for our discussion, the point is that the expression vector you choose needs to be compatible with the host organism and the delivery method that you'll use. Now, let's move on to the next component of expression vectors, the replication. So the origin of replication is going to determine the copy number. So in bacterial cells, copy number means the number of plasmids that are present within a single cell. Keep in mind that more is not always better. High copy plasmids are great for routine cloning and for amplification, especially if transcription is suppressed in those cloning vectors. But for expression, if you've got lots of copies of a plasmid, and that plasmid also contains a strong promoter, then you can easily overwhelm the metabolic capacity of the cell. And if your cells don't survive and grow, then they won't express your target protein very well. So for that reason, most expression vectors use an origin of replication that's sort of low or mid copy number rather than an origin of replication that has very high copy number. On the other hand, for eukaryotic expression vectors, you'll typically have a high copy number origin of replication that's used for amplifying the plasmid in a bacterial host, but then you'll need a different kind of origin of replication in order for the plasmid to replicate in the eukaryotic host. So in eukaryotic cells, plasmids don't normally exist unless they've been brought in by viruses. So there are no naturally occurring eukaryotic origins of replication for plasmids. And a bacterial origin of replication will not allow for plasmid replication in mammalian cells. So instead, eukaryotic expression vectors typically use a viral origin of replication. Sometimes, in addition, you'll see an F1 origin of replication noted on plasmid maps. That will allow for single-stranded plasmid DNA to be produced, which can be useful for certain applications, such as mutagenesis or sequencing, but that's not necessary for replication of the plasmid inside the host or for expression from the plasmid. Okay, so the next component that we'll look at are selection markers or screening markers. Antibiotics are typically used to select for cells that carry your expression vector because your expression vector will contain a gene that confers resistance to the antibiotic. Different antibiotics have different mechanisms of action, which will determine which types of cells are susceptible to being killed by that antibiotic. A few antibiotics commonly used in the lab are listed here. So for example, ampicillin resistance to bacterial hosts can be conferred by expression of a gene called beta-lactamase. So occasionally on plasmid maps you'll see beta-lactamase, but more often it'll just be denoted as AMP-R for ampicillin resistance. The gene for neomycin phosphotransferase 2 is usually abbreviated as NEO, and that confers resistance to several different aminoglycoside antibiotics, including kanamycin, gentamicin, and neomycin. Typically, researchers will select with kanamycin in bacteria, and they'll use an analog called G418 or geneticin in eukaryotic cells. And the single NEO gene works in both cases. But you'll notice there are lots of different antibiotics that are commonly used. And in particular, in mammalian cells, when you want to select for the presence of two separate plasmids, you'll want to make sure that your plasmids contain different antibiotic resistance genes that are complementary, so that you can then apply both antibiotics to select for the presence of both plasmids. In yeast expression platforms, selection is usually performed not with antibiotics, but instead with auxotrophy. So auxotrophy is the inability of an organism to synthesize a particular organic compound required for its growth. So, for example, all yeast will grow on complex media that contains all necessary amino acids, but in order to grow on a medium that is lacking a certain amino acid, the yeast must carry genes that will enable the biosynthesis of that missing amino acid. So for example, there's a yeast strain that lacks the His4 gene, so it requires supplementation with histidine. But if you introduce an expression vector containing the complementing biosynthetic gene, His4, then you can select for transformed cells simply by removing the histidine supplementation from the growth medium. So it's the same concept of selection, where you only allow your transformed cells to survive. In contrast, screening markers will allow untransformed cells to live, but they'll give you a quick and easy way to detect which cells or colonies have been transformed. For example, in blue-white screening, the empty vector will express an enzyme that turns a substrate blue. But if your gene of interest was inserted into the vector, it disrupts the coding region for that enzyme, and so the substrate remains colorless and the colonies appear white. So you can pick the white colonies for further analysis without wasting your efforts to screen the blue colonies, which you know contain empty vectors. In mammalian cells, reporter genes are often employed to easily monitor transfection efficiencies and expression levels. So for example, GFP can be used, or a number of other fluorescent proteins, and those can be used not just by looking at colonies by eye under a fluorescent microscope, but also for fluorescence-activated cell sorting to sort of automate the selection or screening process. Another commonly used reporter is luciferase. So luciferase enables easy quantitative readout with a luciferase assay in which a substrate is added that will be cleaved by your luciferase enzyme to emit light, and the light can be read by a spectrophotometer. So the same, or a luminometer, I should say. This is making use of the same reaction that fireflies use to glow in the dark. So the next component we'll look at are transcriptional promoters. Again, this is not an exhaustive list, but it is meant to convey that you have many options, and it's worthwhile to consider using a different promoter. In fact, a growing trend, especially when you're co-expressing multiple proteins that constitute a pathway, is to create modular or combinatorial libraries that mix and match promoters with coding regions in order to empirically determine which combinations give the highest yield in your system. Transcription, as you know, is regulated both by promoters and by terminators. So your goal is to produce a suitable amount of mRNA so that you can get efficient translation. So you need to figure out which promoter is going to give you optimal, efficient transcription in your system so that you can get mRNA for translation. Now, translation initiation is mediated by the ribosome binding to the mRNA. And translation initiation is often the rate-limiting step in protein expression in bacteria. If the ribosome doesn't bind to your mRNA very often or with very good affinity, you won't get much protein translated. On the other hand, a higher initiation rate does not always lead to higher final protein concentrations. This is because translation initiation is happening concurrently with elongation and protein folding. So if you're translating too fast, you'll end up with a bunch of truncated or misfolded protein that'll either be insoluble or it'll get degraded. So in order to optimize your translation initiation rate, you want to find a sweet spot in the middle that gives you a high yield of properly folded functional protein. I'll talk a little bit later about a powerful tool we have for custom designing ribosome binding sites to help you find that sweet spot. So I'll just say about this, in prokaryotes, the ribosome binding site is called the Shine-Delgarno sequence, and in eukaryotes, it's called a Kozak sequence. There are variations on those sequences that will alter the binding affinity, so that's something that you can tinker with. But just know that ribosome binding has been shown to depend heavily upon the secondary structure of the mRNA and the presence of nearby translation initiation sites that might compete with the intended one, as well as the distance between ribosome binding site and the start codon, and a whole host of other factors. So it's worth pointing out here that you cannot simply treat ribosome binding sites as modular parts the way that you might treat other components of the vector, such as the antibiotic resistance gene, or even the promoter or the terminator. We will come back to RBS later, but to keep moving through our list of expression vector components, let's look at epitope tags, or fusion proteins, and the cleavage sites that can be put between the coding region and the fusion. So fusion proteins can be useful for several different purposes. First, to detect target proteins using antibodies, or to purify target proteins using affinity columns or other methods, or simply to improve solubility of target proteins. Many different tags are available. A few of the most common are listed here. And many expression vectors come with tags already built into either the N or the C terminal, so on one side or the other of the multiple cloning region. Excuse me. Now, keep in mind that a C-terminally tagged construct will allow you to purify full-length proteins. So it might be useful if you're going to be adding a purification tag to put that on the C-terminus for that reason. Sometimes a double tagging approach could be useful. For example, you can put a solubility partner at the N-terminus and an affinity tag at the C-terminus so that you can solubilize and purify at the same time. Tags are generally small peptides that are not expected to interfere with the structure or function of your target protein. However, tags can be removed if you design your expression construct to include a cleavage site placed between the target ORF and the tag. So these are some commonly used proteases, thrombin, enterokinase, factor XA, and TEV. These are and these are the sequences that they recognize. You'll notice, so you can simply encode these cleavage sites into your expression construct in between the enzyme and, in between the tag and the ORF. Keep in mind that, the TEV site here is a little bit longer. That allows it to afford more stringent sequence specificity than some of these other cleavage sites, but all of these are widely used. Okay, so we've gone through all of the basic components of vectors that you'd expect to see in an expression vector map. Now, let's take a look at the most popular expression vectors that are used in bacterial and mammalian expression systems. So the most popular expression system for E. coli is the PET system. This was developed at Brookhaven National Labs, and it's been widely adapted. So there are many different PET vectors available that feature different combinations of promoters, selection markers, and epitope tags. I want to take a moment to talk about the logic that drove the development of these vectors and the key components that make them so effective. So let's think through this. If your goal is to maximize production of your protein of interest in E. coli, you might think to yourself, how can I design an expression vector that will hijack the transcription and translation machinery of a bacterial cell to make it express my protein? Well, it's often useful to turn to nature to see if your problem has been solved already. And in fact, we know that bacteriophages, which are viruses that infect bacteria, are extremely effective at driving high expression of viral protein. One bacteriophage in particular, called T7, carries a very strong promoter and translation signal, as well as a gene encoding a highly efficient RNA polymerase. So the aha moment was, maybe we can take these elements from T7 bacteriophage and put them into our expression system. And in fact, this was wildly successful. The T7 elements could drive very high expression of target protein. The problem is the system is so effective, you can often have your target protein making up 50% or more of total cellular protein within a few hours. This can cause problems for the viability and growth of your host cells. So then we thought, wait a minute, can we put some brakes on this system? Can we control the T7 elements so that we're only expressing the target protein when we want, and maybe not always at maximum level, but instead dictate how much we want, perhaps through dose-dependent induction? Again, we look to nature, and we find multiple ways to regulate gene expression. One is the lac operon. So you're probably familiar with the idea that bacteria can metabolize lactose for energy, but they don't want to waste energy transcribing and translating those metabolic enzymes unless lactose is abundant and other energy substrates are scarce. So they have a sensor that acts as a toggle switch so that when lactose substrate is absent, the sensor will repress enzyme expression, but when the lactose is present, the sensor will permit expression to occur. So the lac operon has been adapted as a control mechanism for pet vectors to allow inducible expression with an analog of lactose called IPTG. So you can add IPTG at the time when you want to turn on expression, and IPTG can be added in a dose-dependent manner to tune the level of protein expression. There are other methods for regulating protein expression as well, such as the use of a T7 lysozyme, which is a natural inhibitor of that T7 RNA polymerase. So you can imagine there are several different systems that have been set up to provide regulatory checks on T7-driven expression. The basic foundational idea here is that you want an expression vector that keeps your gene transcriptionally silent until you choose to induce expression. That's especially important if your target protein could be toxic to the cell. As we mentioned before, toxicity can occur when T7-driven expression might overwhelm the cell's metabolic capacity. But also, some proteins can be toxic even at low concentrations due to the protein's biological effects within the cell. There are special expression vectors that are recommended for use with toxic proteins, which are designed to prevent leaks of target protein expression in the absence of IPTG induction. So pet vectors are the most popular vectors for expression in E. coli, although there are other options for E. coli expression vectors. Now, if you're working not in bacteria, but in mammalian systems, you've probably encountered the pcDNA series of vectors, which are far and away the most popular vectors for mammalian expression. Just like with pet vectors, there are many variants on pcDNA vectors that have different features, but all pcDNA vectors share a few features. They all have a large multiple cloning site, MCS, and you can choose either forward or reverse orientations by looking for the plus or minus in the vector name. They all include the puck origin of replication for maintaining or amplifying the plasmid in E. coli. So that's a high copy origin of replication for amplifying the plasmid in an E. coli host. They also contain the SV40 origin of replication, which was taken from the simian virus, and that allows the plasmid to replicate in eukaryotic cell lines. pcDNA vectors all use the cytomegalovirus CMV enhancer promoter for high-level constitutive transcription, as well as the bovine growth hormone polyadenylation signal and transcription termination sequence, which will enhance the mRNA stability. They also all contain, obviously, a Kozak sequence to enable ribosome binding for translation. The map here shows a version of pcDNA that contains the DYK or flag tag. Other tags are available. And in this case, it's going to be a C-terminal tag, but obviously tags can be placed either N-terminal or C-terminal. Different selection markers are available in pcDNA vectors. So the map shown here shows AMP resistance, which is useful for amplifying the plasmid in E. coli, and a second gene conferring antibiotic resistance in mammalian cells. In this case, it's NEO. But other options are available, such as zeaxanthin or hygromycin. And again, if you're going to transfect more than one plasmid at a time, you want to pay attention to antibiotic complementarity. So for example, you could use this plasmid with neo and another plasmid with hygro, and then you'd apply both of those antibiotics to select only for cells that carry both plasmids. So pcDNA is a very versatile, powerful family of vectors, and it's the most popular for mammalian cells. I don't have time to do an exhaustive comparison of all commercially available expression vectors, and I'm not sure how useful that would be. you can find reference materials for that when you need them. Instead, for now, I'd like to move on to some case studies in customizing vectors. Okay, so case study 1 is about optimizing vectors for CRISPR-Cas genome editing. This first case study is based on a paper that was published by the Zhang Lab at the Broad Institute. You may have heard of that lab for their work on CRISPR-Cas9 genome editing. In this case, they had already developed a lentiviral vector to express Cas9 nuclease and guide RNAs in mammalian cells. But they wanted to optimize the vector in order to improve their viral titers. This was particularly important because they were using large gRNA libraries, the Gecko libraries, which have over 100,000 sequences. So they needed high transduction efficiency in order to limit the vast number of cells required for the screen. So their goal was to improve the viral titer. And in order to do that, they made some modifications. First, their original vector contained 2 nuclear localization signals. Now, I haven't talked about these before, but these are small parts of your vector that will be in frame with the coding region, with the protein that you're trying to express. There will essentially be a tag on the protein that allows the cell to target the protein to a specific region of the cell. So a nuclear localization signal will allow the cell to take this protein into the nucleus, which is what you want for a Cas9, which needs to actually cleave DNA. So it needs to be present in the nucleus in order to be active. So their original vector had 2 nuclear localization signals, but they thought, well, if we want to try to tighten this vector up, make the plasmid a little bit smaller, maybe it'll express better, we could probably afford to remove one of the NLS sequences. So they did that. Then they looked at the expression construct and they saw, we codon optimized the Cas9 sequence already, but we didn't codon optimize the NLS or the P2A bicistronic linker sequence. Now, codon optimization, I'll mention a little more detail later, but it's basically a way to introduce silent mutations that will enable more efficient transcription, mRNA stability, and translation. So they said, we want to go ahead and codon optimize every part of this expression cassette. So they did that. And then finally they thought, well, what if we rearrange the components? They tried a few different rearrangements, and they ended up repositioning the U6-driven sgRNA cassette in a different place from where it had been before. So let's see how their efforts yielded a result. They found 10-fold increase in functional viral titer with all of these changes. So that was pretty good, but they didn't stop there. They thought, well, this vector is still really big. What if we split the components into 2 separate vectors and delivered them sequentially with different antibiotic selection markers? Would that improve our results? Now, normally you would think an all-in-one vector ought to be easier because you just have to get this one construct in and you don't have to worry about selecting for cells maintaining 2 separate constructs. But in fact, they found that with their dual vector system, they observed a 100-fold increase in functional viral titer. So both their improved single vector system and their dual vector system are available for other researchers to use with CRISPR-mediated genome editing. And in fact, GenScript offers CRISPR services where we offer these vectors as well. But this is, I think, a really powerful case where these vectors that were intended as expression constructs for human and mouse cells were able to be optimized just by making a few little changes to the expression vector backbone. Okay, so let's look at a second case study now. Excuse me. This is in bacterial cells. And here, this is a metabolic engineering study where the goal is to optimize conversion of carbon dioxide to butyrate in a Clostridium microbial host. And in this case, the researchers wanted to express not a single protein, but actually 8 different enzymes that make up this metabolic pathway. And so they created initially a strain B1 that expressed those 8 enzymes on 2 separate plasmids. And they noticed that they were getting pretty good expression of 7 of the 8 proteins, but one of the proteins wasn't expressing very well. And so they weren't getting very good output of their pathway. They weren't getting very good butyrate production. So they thought, well, maybe we need to make some modifications to our expression constructs to improve this. And so the first thing they did was they looked at that one protein that wasn't being expressed very well. They saw that it was being transcribed at the same rate as other genes. There was just a bottleneck with translation. And they noticed that the distance between the ribosome binding site and the start codon on that gene was different than for the other genes. So they adjusted the distance between the RBS and the start codon, which seems like a very small change to the expression vector, but we'll see how much of a difference it made. They then went on and said, well, you know, we're expressing these 8 genes on these 2 plasmids that are not integrated with the host genome. What if we tried integrating them with the host genome instead? So they did that using either double crossover or single crossover homologous recombination. And then, you know, they tested the results in that strain. And then they made another modification. They thought, well, instead of simply integrating these 8 genes, what if we made these 8 genes actually replace an endogenous gene that we think may be competing with our pathway. So the PTA gene converts acetyl-CoA into acetate, and they really wanted to divert flux away from acetate production and toward butyrate production. So they thought, let's just knock out the PTA gene, and we'll put our butyrate pathway genes in the place of PTA in the host genome. And that should give us even better flux through our new synthetic butyrate pathway. So let's see what the effect of each of these modifications was. With the first change, they observed a significant increase in CRT protein and a significant increase in butyrate production, the end product of their metabolic pathway. So that was great, even though this seems like a very small change that they introduced. For strain B3, they did observe faster cell growth. So that's consistent with the idea that having the cell maintain these 2 plasmids may be slowing down cellular metabolism. On the other hand, they observed only similar, or in some cases very slightly lower butyrate production. So in terms of optimizing butyrate production, which was their goal, this wasn't really an improvement. To their surprise, perhaps. And then finally, when they knocked out that PTA gene in place of the butyrate pathway genes, they expected to see a lot less acetate and a lot more butyrate, but the effects were much more modest than they had expected. And this was a surprise, and they thought that perhaps you know, this indicates that there are many other ways that the cell can produce acetate, and that the cell has great, you know, biofeedback mechanisms that make it perhaps not as easy as you might think to turn them into factories that are optimized for producing only what you want. So I just want to summarize this case study by pointing out that this very small change in adjusting the distance between the ribosome binding site and the start codon had the biggest impact on helping them achieve their goal of maximizing butyrate production. Okay, so now let's move to part 4, how to optimize protein expression. First, a reminder, the conditions required to achieve optimal levels of active soluble protein are rarely the conditions that would maximize protein expression. Optimal conditions depend on your host cell background, your culture conditions, your vector configuration. In practice, it's usually worthwhile to test several different vector-host combinations in order to optimize yield of target proteins. So we've already talked so far about ways to optimize the expression vector by considering each of the components. And we've learned about some of the variants that are commonly used for each of these components. We saw some case studies where reorganizing components on the vector proved useful. And we talked about RBS design as a powerful method for improving protein expression in bacterial systems, which I'll talk about a little bit more in a few slides. I also want to touch on some other aspects of your workflow that you can optimize. You might want to optimize the cargo, so not the vector, but the insert DNA itself. One way to do that is through codon optimization. You can also do sort of more thorough protein design. And then you may choose to optimize the host, that is the platform or the species and the specific strain that you're using for your expression. And of course, you may consider optimizing your expression and purification methods. So how you transform and select the media and conditions, temperature that you use to grow and to induce expression in the cells, and how you purify the protein from the cells. All of these are steps that can be optimized. So to help with each of these different steps, GenScript has several webinars that are archived on our website for you to view on demand. I'm going to talk briefly in the following slides about codon optimization and about ribosome binding site design. And I would enthusiastically encourage you to check out the webinars by my colleagues on optimizing conditions for soluble protein production and on overcoming some challenges for protein expression and purification. Keep in mind that when you encounter problems, for example, how to promote proper protein folding, there are usually several different approaches that you can take to address that problem at different levels of your workflow. So for example, to promote proper folding, one strategy would be chaperone co-expression. And again, we've got an archived webinar you can check out about that. But as I mentioned earlier, proper protein folding is coupled really with translation initiation and with elongation. And so some other tools that you can use to monitor or to manipulate those two steps are using RBS design to tune the translation initiation rate and using codon optimization to tune the translation elongation rate. So let's discuss RBS design and codon optimization. First, ribosome binding site design gives you tunable control of the translation initiation rate. It's based on a biophysics analysis of all of the factors that influence ribosome binding to mRNA. There's an extensively validated algorithm that was developed by Howard Salas's lab at Penn State University, and GenScript has in-licensed that algorithm to be able to offer RBS design services to our customers as a complementary service. So the algorithm has been cited by hundreds of peer-reviewed papers, and it can be used both for single proteins as well as for multi-enzyme pathways. So the basic idea is that depending on the sequence you want to express, we can look at the sequence and, for example, in the expression vector that you want to use, there will typically be a ribosome binding site already there. We can see what the predicted translation initiation rate would be with your original construct, and then we can design one or more synthetic ribosome binding sites that would have a different predicted translation initiation rate. And so by using this really powerful, very accurate design tool, you can create, for example, a small modular library of different constructs that simply differ in their ribosome binding site that will allow you to sort of systematically explore the entire possible expression space and find what the optimal RBS is for your system. Because even though we can predict what the translation initiation rate will be, we may not necessarily be able to know what the best TIR is for protein expression in your system, since it's all about balancing expression with folding with elongation and folding, balancing initiation with elongation and folding, as we mentioned earlier. So again, this can be used for single proteins or multi-enzyme pathways, and this is a complementary service that GenScript allows. And to learn more, you can check out Dr. Howard Salas's webinar, which is archived on our website. Next, I want to talk about codon optimization. So codon optimization means introducing silent or synonymous mutations into your open reading frame. These do not change the amino acid sequence, but they do favor efficient soluble protein expression by making transcription and translation proceed more smoothly. Codon optimization can be applied not only to your gene of interest, cargo, but also to any linkers and tags that will be translated. So as we saw in one of the case studies, codon optimization of their P2A linker and their NLS tag really made a difference in expressing the entire construct. So it may be worthwhile to synthesize codon-optimized sequences for your tags rather than simply using the tags that come pre-made in commercial vectors. Again, codon optimization does not change the amino acid sequence. Introducing changes in your amino acid sequence can sometimes improve the solubility or the folding of your protein without affecting the critical functions. So this is another strategy that can be employed. We would call this protein design. And again, that's really different from codon optimization. Codon optimization is not going to change the amino acid sequence. And as a final reminder, codon optimization does not alter any portion of your expression vector outside of the open reading frame. So codon optimization does not deal with your promoter or any other aspect of your expression vector outside of the ORF. However, changing those other aspects of your expression strategy independently of codon optimization can lead to synergies. And again, the best way to find this sometimes is by creating a few different constructs or a small modular library of constructs to empirically determine what the best combination is. Regarding codon optimization, it's worth pointing out that tinkering with the codons can improve expression or it can reduce expression levels. So it can be a bit dangerous. And not all codon optimization algorithms are the same. So you do need to exercise some care with this. GenScript's optimum gene algorithm is patented and is regularly updated based on new validated research findings, and hundreds of peer-reviewed publications cite GenScript for codon-optimized gene synthesis. You can search or browse through those papers in our online reference database. You can also read some more case studies in our Gene Synthesis Handbook, which is available for download on our website. When you're ready to get codon-optimized genes for your expression constructs, you can request a free codon optimization report, and then you can order gene synthesis. We offer a few different gene synthesis services depending on how fast you need your genes, how many genes you need, and how long the genes are. Our standard service starts at 35 cents a base pair. The turnaround time is just 8 business days. We also have a chip-based platform that powers our GenPlus high-throughput gene synthesis service. You can consult this chart or our website whenever you're ready to order, or you can talk with a sales rep to decide together on the best services for your needs. I would just point out that unlike some competitors, every synthetic gene that we deliver is cloned into a plasmid and sequence verified. You can choose from our standard cloning vectors at no additional charge, or for a small additional fee, you can request cloning into any vector of your choice. So we are able to offer 77 popular expression vectors at no charge to our customers when you order our express cloning service. This has really been a popular service since it launched last fall. We're continuing to expand the number of vectors we offer. If you don't see your favorite vector on this list, you can always send us an aliquot of the vector that you want to use, and we'll archive it for your exclusive use. So in addition to our express cloning service, we also offer Vector Arc, which offers secure, free storage of your vector plasmids that you send us. This is really helpful for labs where, postdocs and graduate students come and go, and you want to make sure that both the physical reagents and the data regarding all of your plasmids is securely stored and easily accessible by anybody in the lab in years to come. We also offer $49 cloning into your archived vectors anytime you need a new gene insert put into one of your previously archived vectors. In addition, we offer an express mutagenesis service so that if you need multiple expression clones with small mutations, for example, if you want to use our RBS design service, and so you want to then test maybe say 3 or 5 different expression clones that are exactly the same except that they vary in the location of the RBS, that they have different synthetic ribosome binding sites cloned in there. We can use our express mutagenesis service to create those for you, and it's just $99 per mutant clone. We also offer CRISPR gRNA services. So as I mentioned, in some cases, you don't want to drive expression from a plasmid. You want to integrate your expression cassette into the genome of your host cell. CRISPR makes that really easy and very, very efficient to create knockouts or knock-ins. Is the application that I'm mentioning now, as well as other applications. So we've in-licensed technology from Feng Zhang's lab at the Broad Institute so that we can offer all of the vectors that they have developed. And of course, we offer guide RNA design services, and we'll synthesize your guide and clone it into the vector to give you those expression-ready constructs for CRISPR gene editing. So just to back up again and see the big picture of your entire protein expression workflow, I just want to point out that GenScript offers end-to-end solutions to help you at any stage of the protein expression workflow where you might need help. Today, we focused on our molecular biology services, but we also offer numerous downstream services. For example, if you're trying to choose the best host organism to express your protein in, we offer a potential service which will evaluate protein expression levels in small batches across several different expression systems to identify the best host. And then we also offer full service protein expression and purification to deliver any quantity and purity of protein that you need. We know a lot of customers prefer to do their own DNA cloning and protein purification in their own lab until they run into problems, and then they outsource their difficult projects to us. That's great. We love a challenge. So we can express your protein. If you're having trouble expressing your protein, we can do it for you in any of these different expression systems. Further down the workflow, if your purified protein is largely insoluble, you may need refolding, or you may just want some QC, such as MALDI-TOF or HPLC or circular dichroism, to determine the purity and aggregation of your protein. And so we can do a number of different purification and characterization services for you. So I hope today that I have piqued your interest in expression vectors, and I want to let you know that GenScript offers a wide range of custom services as well as catalog products that can help your research. We are the most cited biology CRO, contract research organization. We can create custom bioreagents or perform assays to save you time and to fill in any gaps in your in-house technical expertise or capabilities in any of these areas. So please get in touch if there's ever anything you think we might be able to help you with. Thank you again for attending. I hope you found this webinar useful. Please complete the survey that will pop up on your screen when the webinar ends. If you have any questions or comments on what I've talked about today, I'd love to hear from you. You can email me at rachel.spear@genscript.com. This webinar will be archived on the GenScript website, and the other webinars I mentioned are also available there for on-demand viewing. You can either watch the recorded webinars with audio, or you can simply download the slide decks. And whenever you're ready to request RBS design or codon optimization or gene synthesis of the expression constructs that you need for your protein of interest, you can go to our website and use online quotation request forms, or you can simply email your request at any time to us at gene@genscript.com. So at this point, I will wrap things up and look at any questions that have been submitted and see if we have time for some of the Q&A. Okay, I think, so one question that I see here is, you mentioned expression from plasmid DNA is okay for transient expression in mammalian cells, but can't I also use plasmid DNA for stable expression in mammalian cells? Yes, that's a great question. You definitely can keep plasmids stably expressed in mammalian cell hosts. The only trick is you need to keep the cells under consistent antibiotic selection. And there is a risk here. So if you would, if you'd, sorry, if you happen to, if you keep them under constant antibiotic expression, under constant antibiotic selection, and you find that overexpression of your target protein creates a big metabolic burden for the cells, then cells in which the plasmid acquires a mutation or a deletion in the area encoding the target protein may have a fitness advantage. And so you may end up selecting for cells that maintain a plasmid carrying your antibiotic resistance gene, but they don't actually maintain the gene that's encoding your target protein. So a way to check for that is just always to go back and resequence the insert of the plasmid, and that's where having those sequencing primer sites flanking your insert can be helpful. Again, a strategy to get around that possible risk for stable expression is to simply integrate the gene into the genome. And again, for those, lentiviral vectors are great for, quick and dirty screens and high throughput screens because they're really high efficiency. That's why they're used for those CRISPR vectors. But if you're going to be doing extensive characterization, if you're using a viral system, you're probably going to want to have several different clonal populations that you use and average across to make sure that that any effects you're seeing are not off targets from, off-target effects from where your cassette happened to integrate into the host genome. A way to get around that is to use CRISPR or other forms of targeted genome editing rather than random viral integration. But random viral integration is fine in a lot of cases, and continuing to express from a stably expressed plasmid is fine for a lot of different applications. Okay, and another question is it better to have a tag at the N-terminus or at the C-terminus of a protein? So usually, I think I kind of I touted the advantage of C-terminal tags because they essentially guarantee that you can use the tag to purify a full-length protein, because the C-terminal tag won't be translated unless the entire protein has. So if you've got like a truncation in your protein expression, then the tag won't be included. But on the other hand, N-terminal tags are often advantageous because they provide a reliable context for efficient translation initiation. So the fusion proteins may take advantage of efficient translation initiation sites on the tag. So they can be removed, leaving none or few additional residues at the native N-terminal sequence of the target protein. Most of the endoproteases cleave at or near the C-terminus of their recognition sites. So that's why if you have a C-terminal tag and you're using a cleavage site, then you may end up leaving a few extra residues at the end of your protein, even though you think you've cleaved off your tag. So if you're going to use a cleavage sequence in between the tag and the protein, then having the tag and the cleavage site at the N-terminal side of the protein, of the target protein, can give you a cleaner cut. But yeah, I mean, like I said, there are different commercially available vectors that will have the same tag at the N-terminal versus at the C-terminal, and it's often worthwhile to try both of them and see which works better in your hands. Okay, and I think those are the simplest questions to answer. There was one more I saw that I'll follow up individually with someone on. If you have other questions after hearing this webinar, either live or if you listen to it on demand later, please feel free to email me with any further questions. Thank you again for attending, and best wishes in your research.